In this lecture, we're going to learn about how cells make copies of themselves. So, one of the most important key characteristics that distinguish living organisms from non-living matters is the ability to reproduce or to make something of your own kind, right? So, since cells are the basic unit of life, the continuity of life is based on the reproduction of cells, aka cell division. This is how cells reproduce. So in unicellular organisms, which are organisms that are made from just one cell, they divide in order to make new individuals. And this type of reproduction is called asexual reproduction. So you can see up here an amoeba is the example, and that's for the unicellular organisms. For multicellular organisms, their cells divide so the organism can grow and develop because larger individuals usually have more cells in their body, so they have to go through this process multiple times. Some cells in multicellular organisms also get renewed periodically, so the tissue or organ can maintain their function. One example is that cells line, let's say, the inside of your stomach, your gastric stomach. And our stomach contains very acidic digestive juices that can actually damage our cells. So to make sure the acids don't destroy our stomach, those epithelial cells that line up on the inside of the stomach get replaced by new ones pretty much every few days. So tissue renewal is causing cell reproduction or cell division to go on quite frequently. So how does it actually work? So if we start from one cell, first what's got to happen is that cell's got to get bigger, and then it needs to make an additional copy of its genomic DNA. So it's going to separate the two sets of DNA into two different sides of the cell. And after these DNA separate, the cytoplasm needs to be divided. So it'll be divided into two halves, and then we'll come out with two smaller cells, each carrying a complete set of DNA in their nuclei. And each of these new cells can then repeat the cycle to make even more identical cells, and we just keep going and going and going. This is like the quick and dirty version of cell division. So we'll go through all the different processes involved in this as well. One of the most important tasks during cell division is to pass the identical genetic materials to the cellular offspring, which we call daughter cells. Okay, and it makes sense because an organism is defined by the genetic information it carries in its DNA. So if part of this information is missing during cell division, the daughter cells won't be able to carry out the same function as the parent cell. Imagine if the newly made stomach cell have lost some of its genes and all of a sudden can no longer produce and secrete digestive juices, or maybe an algal cell lost its ability to photosynthesize. It wouldn't be the same as the parent cell. Therefore, a dividing cell must replicate its DNA and allocate two copies to opposite ends of the cell before splitting itself to two to make sure both daughter cells carry the exact DNA the parent cell has. And because replicating and allocating DNA is so important during cell division, let's take a closer look at this genetic material before we move on to more of the details about mitosis, which is the cell division. Okay, so you might recall that all lives carry their genetic material in the form of DNA. DNA are macromolecules made up from nucleotides, and in a typical human cell, that is about 100 microns or micrometers in length, right? It contains around 2 meters of DNA. So that's 20,000 times longer than the cell itself, which sounds like a hot mess to separate, right? And just like we have in the illustration here, it's very difficult to separate two very long entangled yarn strings, but if you organize them into bundles like this, separation becomes much easier. So our cells use a similar approach. Right before cell division, our cells organize our DNA into a condensed structure that we call chromosomes. And this helps separation and allocation of this genetic material during the cell division process. So the way DNA is organized is seen here. In each cell, DNA molecules can be organized into a chromosome, which is this kind of X-looking thing. Okay, so this long string of DNA, which has a double helix structure, is first wrapped around 8 special DNA organization proteins called histone proteins. Okay, so we have these histone proteins, and then that DNA protein complex will further coil into more compact structures like chromatin fibers, or this more condensed chromosome structure. When cells are not dividing, DNA are normally present in the form of long and thin chromatin fibers. So only after DNA has been replicated, chromatin fibers will then further coil and fold themselves to form the condensed chromosomes. And that's a lot easier to separate. The condensed chromosomes are so thick that we can actually see them with a light microscope, as we can see in the figure here, as compared to the chromatin fibers in the neighbor cell. So we can see chromatin here is just kind of like loosely packed in. You have a bunch of different fibers. And then here we can see it's more organized into this condensed chromosome structure that's ready for cell division. So chromosome numbers vary widely among different species. For prokaryotes, most of them only carry one chromosome in their cell, meaning that they only have one DNA molecule, and it's usually in a circular form. For eukaryotes, each species would have its characteristic number of chromosomes. So for example, humans have 46 chromosomes, cabbage have 18, chimps 48, elephants 56, and hedgehogs have 90. So to be a little bit more specific, when we're talking about human chromosomes, the 46 that we actually have are actually two sets of 23. Meaning that there's 23 types of chromosomes in the human body, and each comes in 2 copies. So one is coming from mom, one is coming from dad. This is the case for our somatic cells, which are all of our body cells except for the reproductive cells. Okay? And when a cell carries 2 sets of chromosomes, that's what we call diploid. So you'll see that abbreviated as 2N, so 2 times that number, diploid. Okay, so for human chromosomes, we'll organize them from 1 all the way to 22 based on their size. So 1 is the longest in length and 22 is the shortest. And then our last pair of chromosomes here, you'll see that all of these are numbered, and these are X, and then we have a Y here. So these are the sex chromosomes. Girls would have 2 X's here, and boys would have 1 X and 1 Y chromosome. So female 2 X's, males 1 X, 1 Y. And you'll see these chromosomes here. So homologous, homo meaning same, right? That's the same pair. Heterologous chromosomes are not of the same pair. So if we have one from 1 and one from 2, those would be heterologous, and then both of them in 1 would be homologous. So in comparison to somatic cells, human reproductive cells, or gametes, only carry one set of 23 chromosomes. So these ones are said to be haploid. They're 1N versus our diploid somatic cells. Okay. So during sexual reproduction, 2 gametes, one is coming from mom and one is coming from dad, will undergo fertilization and fuse their nuclei together to get a zygote that again will have 2 sets of 23 chromosomes, 46 chromosomes total. That diploid zygote can then undergo numerous rounds of cell division that lead to the formation of a human baby. So mom and dad get together, fertilize the egg, creates a zygote, and then it goes from a zygote into all the different stages of development until eventually it's the human baby that's being born. Okay, so now that we kind of have a little bit of the background on DNA and the genetic material, let's actually go over the cell cycle. So this is cell reproduction or mitosis, and we're going to see one cell undergoing growth and how it goes into DNA duplication and then division to make two identical daughter cells. Keep in mind, daughter cells doesn't mean that it's female, that's just the context, the name for it. And each of these cells can then undergo the same process to make more cells. So this is what we call the cell cycle. So each cell cycle can be broken into two main phases. We have interphase, and then we have the mitotic or the M phase. Okay, so the mitotic phase is actually cell division, right? And that's only actually a small part of the cell cycle. The cell actually spends the majority of its time in interphase, which contains three subphases. So we'll see here we have the GAP1 or G1 phase, during which the cell is growing and producing proteins and cytoplasmic organelles. And then we go into the S phase, and that's when DNA synthesis occurs. Okay. Then we get into G2 or GAP2, and that's when the cell is growing just a little bit more to help it prepare for that cell division. Okay. There are certain cells in our bodies that do not divide after they're fully differentiated, and they're said to be to stay within this G sub O or GAP0 phase. Okay, so interphase, like we said, is the phase that the cells spend most of their time in, which is basically, you can think of it as the preparatory phase. So this whole blue zone here, this is interphase, and it's just prepping the cell to actually go through the cell division that's all going to happen in M phase or mitosis. So within the M phase, there's two different stages. We have mitosis and something called cytokinesis. So mitosis or karyokinesis, if you want to refer to it as that, but we'll commonly hear it as mitosis. This is the division of the DNA in the nucleus, where cytokinesis is the division of the cytoplasm following mitosis. So when a cell is committed to divide, it enters the S phase in that interphase, that blue phase from the last slide, and that's where it's going to duplicate its DNA. So that happens when the DNA is still in that long fibrous chromatin form. And then once it's divided and it goes through G2, then it'll enter mitosis, right? So we'll see the chromosomes here lining up. We have this thing called a centromere that's kind of holding it together in the middle. And we can see what our chromosomal DNA molecule looks like as we go through this phase. So following DNA replication, that chromatin begins to condense into the chromosomes, as we can see in the figure here. So this chromosome contains two identical copies of DNA, 1 and 2. Right? So these are regarded as sister chromatids. These two sister chromatids are initially attached all along their length, and the region where they're attached most closely, that's what we're calling the centromere. Okay, so each sister chromatid has one centromere, and then we'll get back to what the role is of the centromeres a little bit later, but just important to note, this is sister chromatids, and different than homologous chromosomes that we mentioned earlier, because homologous chromosomes are coming one from mom, one from dad, and these are sister chromatids. They are of the same. Okay, so back to mitosis. So during mitosis, our two sister chromatids will be separated and distributed to two different sides of the cell. So mitosis is usually followed immediately by cytokinesis, which is separating the cytoplasm in halves and results in the formation of those two daughter cells, each carrying the exact copy of DNA as the parental cell it derives from. So this is, I know, kind of a confusing process, and so I do highly recommend watching the Amoeba Sisters video, or you can look up other videos too, but I like the Amoeba Sisters. I think they're very succinct and I like their animation, and it's really easy to follow. Okay, so let's look at mitosis in a little bit more detailed view. So it gets confusing because we have interphase and then we go into mitosis, which contains actually 4 or technically 5 subphases within it. Okay, so these phases are prophase, or sometimes called prometaphase, metaphase, anaphase, and telophase. And then the 5 would come here at the very end with cytokinesis. Okay, so remember DNA duplication is occurring during this S phase, which is then followed by the G2 phase. And during G2, basically the cell is making another copy of the centromere. So you can see the centromeres here. Our chromosomes are duplicated, they're uncondensed in here. And so it's preparing itself for mitosis. So up until G2 phase, that DNA is still in this uncondensed chromatin form. And then when everything's ready, the cell is going to enter stage 1 of mitosis, which is called prophase. So during prophase, chromatin is starting to condense, and then these two centromeres are beginning to move away from each other as they're being pushed apart by these things called mitotic spindles. Okay, and then following prophase, we have prometaphase. So these are basically the same phase. You could call it both prophase, but if we're gonna kind of subsection them, prophase, we're doing this where we're starting to condense the chromatin, and then in prometaphase is where we're going to have this nuclear envelope is now kind of fragmented. So the nuclear envelope that's holding the DNA of the nucleus, right? It's kind of breaking apart now to allow some of those mitotic spindles to invade and attach to the centromeres of each of these sister chromatids from the two sides of the cell. So while other mitotic spindles continue to elongate and push the centromeres towards the pole of the cells. So you'll see here that we're essentially just getting ready. We're all coiled up now, and now we're slowly moving these centromeres in and breaking down the nuclear envelope. So here's a bit more details on how these mitotic spindles attach to the centromeres. So a special protein known as the kinetochore can be found on the centromere of each sister chromatid. And as the mitotic spindle approaches, the two would intertwine to each other, as you can see here in this figure. So this structure creates a very strong hold that allows the spindle to pull the two sister chromatids apart from each other towards the poles of the cell during mitosis. So you'll see them kind of being pulled each way here, and that's happening during some of the next phases. But again, this is what's these mitotic spindle fibers, they're attaching to these kinetochores, those proteins, on the centromere, and then acting to pull those chromatids apart. So back to mitosis, following that prometaphase, now we're going into metaphase. And metaphase, I always just kind of think of it as meta middle, right? So this is where everything is kind of lining up nicely here in the middle. Okay, so the mitotic spindles are now connected to those centromeres of each sister chromatid from the two sides. So we have one connecting on this side, one connecting on this side, and it's going to start moving the chromosomes back and forth until all of them are aligned in the middle, just like as if they were on a plate. So this line where all the chromosomes align together is what they call the metaphase plate. Okay. So up next is anaphase. And anaphase is where we are starting to have these mitotic spindles. And again, they're attached to the centromeres, and now they're beginning to shorten. So as these shorten, right, it's going to pull these sister chromatids away from each other and towards each of the poles. So we're slowly kind of separating those chromatids again and pulling them towards each side. And the mitotic spindles that are not attached to the chromosomes would extend in order to elongate the cell more. So we would have some that are kind of extending on the sides that's helping us elongate more so that we're ready for cytokinesis later. So finally, after anaphase, we're going to enter telophase. And during telophase, that nuclear envelope is beginning to reform. So remember, it was fragmented in the first phase and prophase. Now it's coming back together around the new separated chromosomes while cytokinesis is taking place. So cytokinesis is basically going to cleave right down the middle here and then create two new identical daughter cells. So just a bit more information on how that mitotic spindle managed to pull the sister chromatids towards the pole. So on each of these kinetochores where the mitotic spindle attaches, there's also a motor protein. Motor proteins help to walk the chromosome along the microtubules away from the center. So as the motor protein passes, the microtubule would depolymerize on the other end. So an easy way to visualize this is to think of it as Pac-Man. So as it moves forward, it's eating up the tubule it just passes. So except that this Pac-Man, as it's moving forward and eating up that tubule, it's also attached to a chromosome, and the chromosome would move with it towards the cell pole. So it's like this Pac-Man is eating its way towards one end of the pole, and it's towing behind it a chromosome that it's bringing with it on the journey. Okay, so here's an easy way to remember the subphases. P in prophase and prometaphase stands for prepare, since the cells are getting ready during this time. And then M, metaphase, middle, right? They're all aligned in the middle in that metaphase plate. A in anaphase, it stands for away. So this is when the sister chromatids are being pulled from away from each other. And then finally in telophase, T for two, because this is during that phase where the two daughter cells are formed. So after the chromosomes have been separated and new nuclear envelopes form around the two sets of chromosomes, it's ready for cytokinesis to divide the cell into two. So cytokinesis occurs differently in animal cells and plant cells. And you'll see here we have a visual of an animal cell, and that's where a cleavage furrow is formed near the old metaphase plate right in the middle of the cell. Okay. This ring is formed using cytoskeleton, which just keeps shrinking until it eventually pinches the cell into two. So again, this cleavage furrow you'll see near the old metaphase plate. The cytoplasmic side, you'll have a contractile ring made of actin microfilaments. So this is something that's like found actively in muscles and things too. It's helping it move and interacting with myosin molecules, which causes the ring to contract. Same thing that muscles do that we'll learn a little bit about later in the semester. And so this cleavage furrow is just going to deepen until, again, it's pinched into two. This is within the animal cells. In a plant cell, a little bit different, instead of pinching the cells in half, the vesicles contain cell wall materials that travel through the cytoplasm, through microtubules, and then they arrive at the center of the cell to form this thing called a cell plate. Okay, so they basically deposit the building materials right there to form this cell plate, which eventually extends into a cell wall and then results in the two daughter cells. And a quick peek of prokaryote cell division. Since prokaryotes have no nucleus and normally they just have one circular DNA, mitosis isn't really necessary for their cell division. So most prokaryotes are bacteria in most cases. That's kind of what we're referring to. And they mostly divide by a process that we call binary fission, asexual reproduction. And during this process, they also need to duplicate their chromosomal DNA first, right? And then the cell would grow and elongate, and then it eventually gets divided into two smaller daughter cells. So it's still kind of doing a similar thing, but it's not actually mitosis because it doesn't involve going through all the different steps. It's quite a bit simpler, for lack of a better word, as you go through binary fission. Okay. So now let's learn about how the cell cycle is actually controlled. One thing to know here is that the length of the cell cycle and the frequency of cell division changes from cell to cell. So example, embryonic cells, right, as we're growing essentially what will be a baby, right? They're dividing every few hours. So that's how we can go from one single zygote cell to a human baby within 9 months, is they're dividing very quickly during development. Epithelial cells, which are cells that line your skin, mucous membranes, things like that, they're also dividing pretty frequently. They're dividing every 2 to 5 days throughout your entire life, right? But liver cells, they pretty much only divide when they're needed, and the turnover rate for a liver cell is like 200 to 300 days, 200 to 300 days. So they're basically only turning over to repair injury or maybe to, replace really old cells. And then there's also cells that never divide in our body. Nerve cells and muscle cells don't divide. They're stuck in this initial G sub O phase in mature humans. So that's why muscle injuries are really difficult. Neuronal injuries, any sort of nervous system injury is usually irreversible because of that, because you won't be able to make those cells again. And the reason why all these cells have different division frequency is because their cell cycles are regulated at a different molecular level depending on what cells types they are. So let's talk about these things called checkpoints. This is how a cell cycle is controlled. And basically these are various signals that are used to determine if a cell is going to pause or if it's going to continue progressing to the next phase. So a cell will stay at its current stage when it receives a stop signal. And it's only going to move ahead to the next phase after receiving a go-ahead signal. So the reason for checkpoints is they can prevent compromised cells from dividing and being able to pass that air to its cellular offspring. So you'll see different checkpoints that happen here throughout the whole phase that allow this to be checked. Because the cell carries out different tasks during the different phases, all of these different checkpoints check for different things. So for example, during the G1 checkpoint, this checks for all different conditions, external and internal, as well as DNA damage. So for the most part, I kind of think of this as DNA damage, right? Because if we have damaged DNA, we don't want to go on to the next point where we're going to replicate it. So we're looking to see if we need to fix or pause it and enter into G sub O. Okay. If the DNA looks good, then we're going into S phase, and that's where we're going to synthesize or duplicate our DNA. But once we do that, we want to first see if it duplicated successfully. So we have an S checkpoint as well that's going to check and see if it duplicated successfully and if the DNA is damaged. And if it's not, then it can continue to go on to the next point in G2, right? But we don't want to pass damaged DNA because we want to make sure that we're passing correct genetic material that's crucial for cell division and normal cell function. So if everything's normal here, we pass this checkpoint, then we go into G2. And during the G2 checkpoint, the cells are going to check if all the sister chromatids are attached to the mitotic spindle correctly. So this way it ensures the accurate separation of the sister chromatids during the later anaphase so that both daughter cells end up with a complete set of chromosomes following the cell division. So now that we know that the cell cycle is controlled by these things called checkpoints, let's take a look at different types of signals that control these checkpoints. So the first type are the chemical factors such as things like growth hormone, right? So for example, when a cell is damaged and dying, it would release growth hormone to its nearby neighbors and signal them to divide and make new cells in order to replenish the spot. So another example, like say during an injury, platelets in our blood would release PDGF, which is a specific platelet-derived growth factor, okay, in the vicinity to stimulate fibroblast cells to divide in order to heal that wound. Right? So these growth factors are usually used as a go-ahead signal within cell cycle regulation. The second type of signals are the physical factors. Okay, so for example, when the cell density is too high in a tissue or culture flask, their surface markers would touch each other, and that would make them stop growing. So high density will serve as a stop signal in a cell. Okay. The other one is called anchorage dependence, and basically what happens is many cells can only divide when they are attaching to something, and so if they're free floating, they're not going to be able to divide. Therefore, anchorage is used as a go-ahead signal for these cells as well. In addition to the various chemical and physical factors, there's also a specific group of molecules that are used by the cells to regulate cell cycle. So just like the stop and go ahead signals, some of these regulator molecules are positive regulators, while others are negative regulators. So for positive regulators, their presence signal the cells to move forward in the cell cycle. So examples of positive regulators are go signals, are things called cyclins and cyclin-dependent kinases, CDK. Okay, and these basically just need to reach a certain concentration in order to drive the cell cycle forward. The negative regulators are things like retinoblastoma proteins, P53, or P21. And these ones are the stops. So we had the positive was the go, the negative is the stop. And these guys, for example, P53, is a really important cell cycle regulator. So it's responsible for detecting DNA damage at G1 and to halt the cell cycle to recruit repair enzymes in order to fix the problem. So if the damage is too severe and it can't be fixed, P53 here is going to induce apoptosis. Remember, that's cell death or self death basically. So apoptosis will destroy the cell so that it cannot pass the air in its genetic material to the cellular offspring. So if it's working normally, it's going through this, right? It's saying, okay, P53 will say, stop, we need to repair, or, okay, we cannot repair, we need to go into programmed cell death, apoptosis. However, if your P53 is mutated, right, it won't recognize that there's that DNA damage, and the cell cycle is just going to continue. And if the cell cycle is continuing and continuing to duplicate mutated DNA, mutated P53, etc., that's when cells can become cancerous. That's essentially what cancer is. It's unregulated cell division. So what happens in cancer is that cancerous cells are skipping these cell cycle checkpoints, and they're not repairing things, and they're continuing to divide. And those are the cells we don't want dividing, are the ones that don't have the stop proteins, or or maybe the other way too, but they're the ones that have damage and are continuing to go without checking it and going through apoptosis if necessary. So the regulation of cell cycle is very crucial to our health. Cells really only should divide when needed, and that's why we have all these different mechanisms to regulate the cell cycle. When it loses control, it could be because our cells stop responding to normal signals that regulate the cycle, or it could be dysfunctional positive or negative regulators. Then the cell is going to divide regardless of external and internal conditions. So these are our cancer cells, right? They're not going to respond to those normal signals that would regulate the cell cycle. They don't require growth factors to grow and divide, and they don't exhibit either density-dependent inhibition or anchorage dependence. So you'll see here our normal cells, we add growth factor, or maybe, we add growth factor, they're dividing. We don't have growth factor, there's no division. Cancer cells, with or without it, you're still going to see them dividing. They're still going to be going through that cell reproduction without going through the cell cycle checkpoints, essentially. And so they're going to be creating cells that we don't want to have. The consequence of uncontrollable cell cycle is that formation of cancer. So when a single cell loses control of its cell cycle, it can be converted into a cancer cell, and eventually that can proliferate and form a tumor.